Yes. So we saw the structure of mitochondria. Very gross. Briefly, we saw the structure of mitochondria. So it is a membrane-bound organelle, right? Cell organelle. It has a lot of projections of the inner membrane inside the cavity. That those are called cristae. And we also discussed that mitochondria, since the mitochondria is powerhouse. And what do we mean by that powerhouse? Why is it called powerhouse? Because it produces a lot of ATPs. And since ATP is also a chemical chemical compound that cannot be produced with the with a with a magic wand, right? Through some chemical reactions, ATP has to be synthesized. And now we are going to exactly do this. Look at that reaction, which gives us ATP. ATP using some oxidation reactions. Like I said that there are two coenzymes in organelle, NADPH and FADH2. The oxidation of these two compounds is highly exergonic. In fact, they release about 53 and 46 kilocalories per mole respectively, right? So it's a lot of energy to synthesize ATP molecules. Now let us see how this happens. Okay, so the topic of today's discussion is how the oxidation of NADH2 happens. FADH2 happens, and how the oxidation of NADH happens. And how the energy released due to these oxidation reactions, because any oxidation is an exergonic reaction, is coupled with ATP synthesis. Fine. Fine. Now we come to this concept called electron transport chain. In fact, the complete structure of electron transport chain is very extensive. It's very complicated, in fact. But at a basic level of discussion, what we are doing, we just require some important components of electron transport chain. That's it. So let us see, first of all, where is the location of this electron transport chain? The electron transport chain is located, see this, the electron transport chain is located in the inner membrane of mitochondria. Okay, so this picture, the membrane of mitochondria is expanded here. Can you see this? This is a schematic representation of mitochondria, and a small portion of the membrane is expanded. And like any cell membrane and organelle membrane, the membrane is nothing but a plasma membrane. Even the cell membrane is plasma membrane. Similarly, the organelle's membrane is also plasma membrane. You have that lipid bilayers, that's a membrane. So the electron transport chain is nothing but a set of membrane bound proteins or membrane bound protein complexes. So I hope you know a gross structure of cell membrane. A cell membrane is not uniform, right? It is not made up of only lipids, bilayer of lipids. It also contains a lot of embeddings, lot of things which are inside this membrane, a lot of proteins, glycoproteins. You have something called ion channels, as well as phospholipids, receptors. What are these receptors? Receptors are nothing but special proteins which are on the cell membrane. And electron transport chain is also similar to that. It is a collection of proteins, closely lying proteins which are neighboring to each other, and is responsible for some catalysis function, enzyme functions. The complexes of this electron transport chain are this: the complex 1, complex 2, complex 3, 4, and ATP synthase complex. How are these complexes named? The complex 1 is called NADH dehydrogenase complex. Okay. Okay, fine. NADH dehydrogenase complex, the complex 1. The complex 2 is called the complex 2. Yes, the complex 2 is called cytochrome BC1 complex. Complex 3 is cytochrome AA3 complex. Fine. Now, what is a complex first of all? We know the other meanings of the word complex, right? We usually go to shopping complexes. So what's a complex? Complex is a set of something in a shopping complex. Why is it not called a shop? Single shop. Because in a shopping complex houses a number of shops of different kinds. So even a complex in biochemistry means it is not a single protein. A complex is a collection of many molecules. Importantly, proteins, of course. Any complex chiefly contains proteins. But along with that proteins, there are other organic molecules, metals in the complexes. Therefore, if we look at NADH dehydrogenase complex, the coenzyme inside that complex, is FMN, flavin adenine mononucleotide. FMN is the coenzyme inside the complex. Now, if we recall a bit of enzyme structures, right? Usually the enzymes for their function require other molecules also. And these other molecules are called either cofactors, coenzymes, prosthetic groups, or metal ions. Fine. So without these helping molecules, the enzymes don't do their catalysis function. So a complex is usually this. It contains an enzyme along with cofactors, coenzymes, metals, all these things. So the complex 1, which is called NADH dehydrogenase complex. The name of the complex is NADH dehydrogenase complex. The cofactor is FMN, flavin adenine mononucleotide. And in complex 3, it is also called cytochrome BC1 complex. What are the cofactors? They are cytochromes. Okay. So what are the cytochromes? Cytochromes are metalloproteins. Please remember, cytochromes are metalloproteins. They contain two metals, iron as well as copper, basically. Although it's the iron which is which is an important carrier of electrons, they also contain copper in that, Cu and Fe. And the cytochromes are not homogeneous. In the fact, you can identify many types of cytochromes. Like, you know, a pigment, chlorophyll, is not homogeneous, right? There are many subdivisions in chlorophylls, chlorophyll A, B, like that. Xanthophylls, those are all pigments which are broadly classified as chlorophylls. Similarly, cytochrome is also a pigment, generally called cytochromes, but there are a lot of classification within that. A, B, B1, C1, like that. A3. These are subdivisions of cytochromes. But anyways, we will not go into the importance of these structural differences. That's fine. That's what, as I told, electron transport chain is too much to learn. We are just taking only a tip of the iceberg, yeah, basically. When we are looking into this electron transport chain in your biochemistry syllabus, it is only a tip of the iceberg. Still, there is a lot of research going on in understanding the complete structure of electron transport chain and the role of every molecule or every compound in that electron transport chain. Still, a lot of research is going on. In fact, new results refine the old ones. Sometimes new results discard the old ones. Like that, a lot of the models are still being built. Electron transport chain models are still being built. Anyways, we'll just take only just essential for our discussions topic. Fine. Now, why is it called an electron transport chain? This is the important question. Because it transports electrons. Now, when we tell it transports electrons, from where to where? It transports electrons from where to where? It's a very important question. And here lies the understanding of our oxidative phosphorylation. Okay. So, when we tell that NADH is oxidized and NADH is oxidized and the oxidation results in so much energy, minus 52.6 kilocalories per mole. What do we mean by this oxidation? Look at this equation, NADH. The oxidation is always the removal of electrons. Not always. Removal of electrons is also oxidation. And these are the other products of oxidation, H plus and NAD plus. So, these electrons, in this equation, what is the problem? The equation which I have written, I have not shown you who is oxidizing NADH, right? I've just seen a reversible equation where NAD plus can be reduced to NADH, NADH can be oxidized to NAD plus. But look at this equation, NADH, this the reverse direction I have expanded here, plus H plus oxygen. So who is accepting electrons the NADH is giving? Oxygen. Fine. So when you allow the NADH and oxygen to directly react with each other, this happens in a single step. Simply the electrons are removed from NADH by the oxygen. Why is oxygen is a oxidizer? Because it has tendency, very big tendency to grab electrons, to accept electrons from different substrates, from different compounds. So oxygen, molecular oxygen is an oxidizing agent. In fact, it's a very, the very readily available oxidizing agent, very inexpensive oxidizing agent, oxygen in the air. So that's what happens. The electrons are directly transferred to oxygen, but then that becomes combustion. That's what I told. So if this happens in a single step, what happens in a single step? The transfer of electrons from NADH to oxygen happens in a single step. It becomes combustion. So at a shot, there is a burst of energy which cannot be conserved, which cannot be transduced, converted into a useful form of in the form of ATP chemicals. Therefore, like we discussed in our previous class, the oxidation happens stepwise. So NADH cannot transfer its electron directly to oxygen. Then that will be the one-step reaction. Therefore, NADH transfers the electrons to something else. And that transfers to one more carrier. So there is a series of carriers. And the final carrier transfers the electrons to oxygen. That's what the electron transport chain is. The flow of electrons which are liberated from NADH to oxygen through many carriers in between. So that is what the electron transport chain is. So it is transporting electrons from where to where? From NADH to oxygen. Okay, so let us follow the path of this electron. How is it transported? As always, the NADH is there. That becomes NAD plus basically. I have not shown plus here. It's okay. NAD also is fine. So it liberates an electron. And who accepts that electrons? The NADH dehydrogenase complex 1. And there is a coenzyme there, which is called FMN. And what happens to FMN when it accepts electrons? It becomes reduced. It becomes reduced to FMNH2. But this doesn't stop here because the electrons have not reached oxygen yet. They are now in FMN. FMN again becomes FMNH2, again becomes FMN, and releases the electrons in this oxidation reaction, which is taken up by a carrier called CoQ, coenzyme Q. Like I showed you, these are the complexes, complex 1, 2, 3, 4 and all. And there are carriers in between complexes. So what are these carriers? Because basically these carriers are labile proteins. Okay. So the proteins which are these membrane proteins can be classified into some types. For example, there are proteins called transmembranal proteins, which extend from one end of the membrane to the other. There are proteins which are fixed to the membranes. There are proteins which are labile in the, labile means floating in the membrane. So carriers are labile proteins which are there. Coenzyme Q is a carrier between complex 1 and complex 3. Please remember, we are not discussing complex 2 as of now. We have kept it aside. So where is the electron now? In the complex 1. Now it has come to complex 3 through coenzyme Q. So coenzyme Q is a carrier. Fine. So coenzyme Q, when it accepts electrons, becomes CoQH2. That releases electron. And the electrons which are released goes to complex 4. From 3 it goes to complex 4. From 4, within complex 4, there are lot of cofactors which accept and release electrons alternatively. So what are these cofactors? You have cytochromes B1, B, C1, C, A, A3. And every time a compound accepts electrons, the metal ion in it becomes reduced. When it gives electrons, it becomes oxidized. So you should also learn to write this sequence of electrons. So what is that sequence of electrons which you have to learn to write? Show the electrons paths perfectly. Okay. So starting from NADH, how the electrons travel to oxygen. They go to FMN. From FMN, they go to coenzyme Q. From coenzyme Q, they go to cytochrome B. And cytochrome B contains metal atom, iron. When the electrons are accepted by the iron, it becomes Fe2+. When the electrons are transferred to the next complex, the iron becomes Fe3+. So what is the importance of metal ions in these cytochromes? They are the electron ferries. One important property of these transition metals, like iron. Iron is a transition metal, even copper is. They exist in stable variable oxidation states, right? You have two stable oxidation states for iron, Fe2+ and Fe3+, as well as for copper, Cu1 and Cu2+. So the nature has made use of this property of this transition metals, the variable oxidation states. So once the transition metal ion accepts electrons, it goes to its lower oxidation state. When it liberates electrons, it goes to its higher oxidation state. The higher oxidation can accept electron, the lower oxidation state can release electrons. So you can alternate the metal ions between these two oxidation states of its. So what can be done? The metal ions can be used as electron carriers. So when you assemble a series of metal ions, you can transport electrons from one molecule to the other. So that is the reason even the catalysis property of metal transition metals. They are good catalysts also. Why? Because they exist in this variable oxidation states. They accept electrons from one molecule, from the substrate A, and give it to some other substrate B, like that. The same thing happens here. Almost the same thing happens here. So the electrons are carried through cytochromes, and see how many cytochromes are involved in the electron transport. 5 cytochromes are involved. And which are those 5 cytochromes? Cytochrome B, C1, C, A, A3. Fine. Okay. And now focus on the last site of the electron. Where is it now? It has come from complex 1, that is from NADH, and has reached the last complex, complex 4. What happens after complex 4? Where the electron goes? The electron from complex 4, from the cytochrome A3 of complex 4, is finally transferred to oxygen. So the O2 becomes H2O. If the oxygen gets electrons, it becomes an oxide ion. Immediately it combines with water, H plus, not water, H plus in the cytosol, in the fluid around it, becomes water. So what is the reduction product of water? Oxygen, water. So you can reduce oxygen to water. That's what happens. But please note that the oxygen is not reacting with NADH directly and oxidizing it. Rather, it is reacting with cytochrome A3. Where is NADH and where is cytochrome A3? They are all interconnected by this electron transport chain. Therefore, the complex 4 is also called, the other name of complex 4 is cytochrome oxidase. Cytochrome oxidase. Why? Because it directly reacts with oxygen. And oxygen is always called the terminal electron acceptor of ETC, electron transport chain. The terminal electron acceptor of ETC. Okay. So what is the net reaction of ETC? What is the net reaction of ETC? Okay, because since I have just explained you these things, you can now tell what is the net reaction of ATP. Yeah, sorry, what is the net reaction of ETC? Net reaction of ETC. Someone can tell? So what has happened because of the flow of electrons through ETC? What has happened? What is the overall result of ETC? Anyone can answer this? What is the overall result of ETC? Yeah. Someone? If you have understood this, you can answer, right? What is the purpose of ETC? At least you answer what is the purpose of ETC? What's the purpose of ETC? Somebody? Should I call out the names and ask the question, or you can voluntarily answer this yourself? Yeah. What's the overall result? I've been talking of this since 2 classes, yeah? And you means for the answers, then okay, let me specify.
NAD plus H2O.
What?
NAD plus H2O.
NAD
plus H2O.
That's the product. Correct. NAD plus H2O. What's the reactant?
NADH.
See, who is answering? Yeah.
Plus H plus Tejaswini.
Yes, go ahead.
NADH plus
O2, oxygen. Yes.
Plus H plus.
Yes, correct. So that's what I meant.
Gives rise to
NAD plus water.
H2O.
Yes. Correct. So that net reaction is same. Is it not? Nothing else has happened. Has any other molecule got oxidized or reduced in the whole process except NADH?
ADP is oxidized to ATP.
No, Tejaswini, we are not still. And by the way, if ADP has to convert it to ATP, oxidation should happen or phosphorylation should happen?
Oh, sorry, sir. Phosphorylation.
Yeah, phosphorylation is not oxidation. Okay, so don't bring ATPs into the picture at this point. So what is my question is, see, there are many compounds here, right, in the electron transport chain. FMN, coenzyme Q, cytochrome B, C1, CA3. Has any other compound got oxidized or reduced apart from NADH? Has any other compound got oxidized or reduced?
FMN2, FMNH2.
H2, yes, that should happen. But when FMNH2 releases electrons, it goes back to FMN. Do you understand? Similarly, coenzyme QH2, it accepts electrons, becomes QH2, but it releases electrons and goes to Q. So what's the point? No other molecule is oxidized or reduced except NADH2. Instead of directly giving it electrons to oxygen, it has given it to oxygen through carriers, through transporters. Do you understand this? So that's what the working of ETC is. Fine. So that's the point. So now we haven't discussed this complex 2 yet. Complex 2. Complex 2 is called, I told you, which are the two coenzymes that get oxidized? One is NADH and the other is FADH2. Electron transport chain is responsible for both the reactions, for oxidation of NADH as well as FADH2. And what is the site of ETC where FADH2 gives its electrons? What is the site where NADH gives its electron? Complex 1, the NADH dehydrogenase complex. What is the site where FADH2 gives its electron? That is complex 2. Complex 2. Fine. So the name of this complex is succinate dehydrogenase. Okay. So why is it called succinate dehydrogenase? That we will understand when we take Krebs cycle. But anyways, you just understand this much. In the complex 2, FADH2 is oxidized. Okay. FADH2 is oxidized. But the electrons from complex 2 goes to complex 3 only. So complex 3 accepts electrons both from complex 1 as well as complex 2. Both from NADH dehydrogenase as well as FADH2 as well as the succinate dehydrogenase complex. From here onwards, the journey of electron is common for both the oxidation reactions, NADH or FADH2. So that's what happens. But fine. Till now, we have been talking a lot about this ETC and we are concentrating on the oxidation of NADH2. But what is the purpose of oxidative phosphorylation? Is the synthesis of ATPs, right? That point we have not touched at all till now. And nowhere in the electron transport chain, the ATPs are coming into picture. So in the next class, we are going to answer this question. How is the synthesis of ATP connected to ETC? Because ETC, we have seen ETC is a very important machinery for any cell because it oxidizes two coenzymes which are highly exergonic, that is NADH and FADH2. But the energy is liberated, of course. But how is that energy used to synthesize ATP? Okay, so for that, we will look into one more interesting aspect of ETC in the next class. Fine. So I'll just stop here. You